In this video, we will discuss the pathogenesis of breast cancer. The word pathogenesis means development of pathology. Now, for the pathogenesis of breast cancer, there are three factors that interplay to result in breast cancer. These three factors are genetics, hormonal effects, and environmental factors. These three factors interplay to result in the pathogenesis of breast cancer, and we will discuss each three of these in detail. So, the first and most important thing is genetics. You know that cancers are caused by mutations in the genes, and these mutations can either be inherited. In that case, we label the breast cancers as familial breast cancers, or they can be sporadic. Sporadic means that mutations are not inherited at all. Rather, the person acquires mutation throughout her life by random chances. These are called sporadic mutations. And most of the mutations in the gene that lead to breast carcinoma are sporadic. But the greater understanding of pathogenesis of breast carcinoma comes from those mutations that are inherited. So we will discuss the role of genetics. We will discuss what are the breast cancer susceptibility genes. Now, firstly, let's understand the point that when we use the word inherited mutations in the context of breast cancer, then that does not mean that a woman had breast cancer, she went pregnant and had a baby, and the baby also inherited that breast cancer. In the context of breast cancer, this is not the case. Rather, in breast cancer, the word inherited means that the newborn have increased susceptibility to breast cancer because they are born with inherited mutation in one of the genes. Now, to clarify this further, you have to understand the concept of tumor suppressor genes. You know that each of the genes present in our genetic code has two alleles, two corresponding alleles on two corresponding members of one chromosome. So, for example, this is a pair of chromosomes. Then, on the same locus, on the similar loci, there will be two alleles of a gene. Now, you know that in case of tumor suppressor genes, both of these alleles need to be mutated in order to result in the disease. Now, you must recall the two-hit hypothesis of breast cancer. Two-hit hypothesis means if one allele is mutated, it will not result in disease. Rather, for the disease to happen, there must be two hits. Two hits in a sense that both alleles must be mutated in cases of tumor suppressor genes. Now, inherited breast cancers occur in a way that a person is born with one of the defective allele by default. So, it happens that during the formation of zygote, one of the allele undergoes mutation. Now, even if the one allele undergoes mutation, it does not immediately result in breast cancer. But that person is at greater risk of breast cancer throughout her life. Why is it so? Because now she only needs one hit to mutate the normal allele. A normal person would be different in that regard, because a normal person would need two hits on the two alleles to be mutated. But in a person who is born with defective allele, then she will need only one hit to make her susceptible to breast cancer. So we can say that if one of the alleles is defective by default, then the person will have increased susceptibility to breast cancer. These are called inherited or familial breast cancers. So I hope that you have now understood the concept of familial breast cancers. That familial breast cancers are those in which a person is born with one defective allele of a tumor suppressor gene, and if the second allele undergo mutation throughout the life, then this will drive the process of breast cancer. Now, what are those susceptibility genes that result in breast cancer? There are 5 to 6 of these which you need to remember. The first and most important and most common are BRCA1 and BRCA2. And remember the very important point that these two genes, if they undergo mutation in both of their alleles, they result in increased risk of not only breast cancer, but also serous ovarian cancer, prostate cancer, and pancreatic cancer. So you have to memorize the names of the cancers that can be caused by mutated BRCA1 and BRCA2. These are breast cancer, serous ovarian cancers, prostate and pancreatic cancers. Now, BRCA1 gene is present on chromosome number 17 on its long arm. Q means long arm. So chromosome 17 Q21 means that it is present on the long arm of chromosome number 17, while BRCA2 is present on 13Q, which means on the long arm of chromosome number 13. Now, BRCA1 is associated with triple negative breast cancer that are known as basal-like cancers, and you know that these are the most aggressive cancers. So remember, BRCA1 results in triple negative cancers usually, and mutations in BRCA2 usually results in ER positive cancers that are less dangerous as compared to triple negative. So this is an important MCQ bundle. Remember all these points. Now, if a person is born with one defective allele of this BRCA1 or BRCA2, then we know that that person will be at increased risk of breast cancer. To actually undergo breast cancer, we need two defective copies of the alleles. But if a person is born with only one defective allele, then even though there will be no breast cancer, but the person will be at increased risk of breast cancer. And if we detect it somehow, we have to do something to prevent breast cancer. And the preventive management of BRCA1 and BRCA2 mutations is mastectomy, which means removal of the breast. Why we remove the breast? Because when there is no breast, there will be no risk of breast cancer. And we do salpingo-oophorectomy. Salpingo-oophorectomy means removal of fallopian tube and ovaries. Why we are removing these tissues? Salpingo-oophorectomy, fallopian tube and ovaries. Because BRCA1 and BRCA2 mutations not only result in breast cancer, rather they result in serious ovarian cancer which arise from the ovary and fallopian tubes. So in order to prevent the chances of cancers in a person who has inherited one defective allele of BRCA1, we need to undergo mastectomy and salpingo-oophorectomy. Now another important gene in the development of pathogenesis of breast cancer is TP53, which results in a protein P53. P53 is also a tumor suppressor gene. So if a person is born with one defective allele of P53, then she will again at increased risk of breast cancer. And usually in the cases of P53 mutations, triple negative and HER2 positive tumors are more common as compared to ER positive tumors. And the syndrome in which P53 mutations are inherited are known as Li-Fraumeni syndrome. Another important allele is CHEK2, another is PTEN. The syndrome in which there is inherited defect in this gene is known as Cowden syndrome. The last gene whose defect make it more susceptible to breast cancer are ATM. The defect in one of the alleles of ATM gene result in a syndrome ataxia telangiectasia. So you just have to remember the name of all these genes and a little bit more detailed about these two genes. BRCA1 and BRCA2. Other genes are P53, CHEK2, PTEN and ATM. Now other than these genetic tendencies, the second effect is that of hormones. And for hormones, we have already discussed in the introduction and risk factor section that estrogen causes release of some growth factors that increase the chances of tumor development and progression. So remember, all the risk factors that cause increase in estrogen exposure result in increased susceptibility to breast cancer. And the third is the role of environment. Of course, we have discussed that heavy alcohol drinking is associated with breast cancer, obesity is associated with breast cancer, and some chemicals like organochlorine pesticides are related with breast cancer. So these three genes, hormonal effects and environment interplay to result in the pathogenesis of breast cancer. So after we have discussed the major factors that interplay to result in breast carcinoma, we have to study the major pathways that lead to the pathogenesis of each of the three biological types of cancer. You know that we studied there are three biological types of breast cancer: ER positive, that are also known as luminal, HER2 positive and triple negative. And there is a genetic pathway of progression or each of these. So let's start with ER positive HER2 negative tumors that are called luminal tumors. Now first see this, that each of the invasive carcinoma types is preceded by a precursor lesion, ductal carcinoma in situ. It is true for ER positive tumors, it is true for HER2 positive tumors, and it is also true for triple negative tumors as well. But the initial precursor lesions are slightly different for each of these. So let's discuss. So for luminal tumors or ER positive tumors, see this example, that for example if this is a normal breast, firstly what happens, that few of these breast epithelial cells become atypical in their structure. Why they become atypical? Of course due to mutations. If this happens, it is known as flat epithelial atypia. Now after this, these atypical cells start to divide and result in atypical ductal hyperplasia. Initially it was flat lesion because there was only a single layer of cells, but now as the atypical cells start to proliferate, it results in hyperplasia known as atypical ductal hyperplasia. Then the atypical cells become so dominant in their quantity that we call it as ductal carcinoma in situ, and after that ductal carcinoma in situ transforms into the invasive breast cancer. So the precursor lesions are normal breast turning into flat epithelial atypia, which results in atypical ductal hyperplasia that leads to ductal carcinoma in situ, and ultimately it results in invasive carcinoma of the breast. Now, what are the genetic disturbances in each of these steps? Remember that we are studying ER positive tumors. So for ER positive tumors, when normal breast converts into flat epithelial atypia, the usual culprit mutations are germline BRCA2 mutations. Remember that ER positive tumors are associated with BRCA2 mutations, while triple negative tumors are associated with BRCA1 mutation. This is an important MCQ point. So the culprit mutation is BRCA2 mutation, and other than this there is loss of chromosome on chromosome number 16 and gain at 1q. Q means long arm of chromosome. So 1q means long arm of chromosome 1 and 16q means long arm of chromosome number 16. So 1q gain, 16q loss, and BRCA2 mutation are the culprit mutations that result in flat epithelial atypia. After that, when flat epithelial atypia converts into atypical ductal hyperplasia, the culprit mutation is PIK3 mutation. So these are the common mutations that have been shown to result in ER positive tumors. After that, let's see HER2 positive tumors or HER2 positive tumors. In HER2 positive tumors, the initial precursor lesion is atypical apocrine adenosis. It is also a proliferative breast lesion which converts into ductal carcinoma in situ and then into invasive cancer. And the culprit mutations in HER2 positive tumors are HER2 amplification, obviously that is why it is known as HER2 positive tumor, and germline P53 mutations. Now the third category is triple negative or basal-like tumors. In this pathway, normal breast converts into some uncharacterized precursors that have not yet been very well understood, which leads to ductal carcinoma in situ and then invasive cancer. In this, the culprit mutation is BRCA1 mutation, and at the later stage of pathway, there is P53 mutation or BRCA1 inactivation. So these are the common mutations in each of the pathway. These diagrams are drawn in Robbins pathology and I have simplified all these. So remember these three pathways.